Hello everyone watching this class. Today in this lecture we are going to study about the very first topic known as artificial hybridization. So first we will start with artificial hybridization. In the last lecture we had studied about pollen pistil interaction, and now we are starting with artificial hybridization. Now what is artificial hybridization? See, this is the technique in which the desired, if we want some desired desired features in a plant, or whatever features we want, like suppose you found this pollen, and this pollen grains of one plant was very useful, it was giving good variation and all, so you collected these pollens. So these are the desired pollens which were giving you desired characters. So whatever, like which the pollens which are desired pollens, those pollen grains are actually collected. Okay, those pollen grains are actually collected and they are dusted upon a flower. Why they are dusted? Why we are collecting the desired desired pollen grains? For getting good amount, like good quality of flowers. So these are desired pollen grains. So I took desired pollen grains which were actually, what they are doing, they are giving good variation, or they are giving good quality of plants. Good, or you can say, instead of saying good, you can say superior quality of plants. Okay, superior quality of plants. So I took these pollen grains and I dusted on the stigma. Where I dusted on the stigma. Okay, this is a flower, I dusted on the stigma. Okay, so it will come here. These desired pollens will come here. Okay, so this is thing known as artificial hybridization. But we have some steps to be followed during artificial hybridization. Okay, so what are the steps? Let's discuss the steps. So steps for artificial hybridization. The very first step is, like, you have to bring pollen grain. Okay, you have to bring pollen grain, means collection of pollen grains. Collection of pollen grains. Okay, then second, you have to take a flower and you have to do emasculation procedure. Emasculation means removal of anther. What is emasculation? Emasculation means removal of anther from a particular flower. Okay, I will show you the image. See, in this figure you have female part also and male part also. Okay, you have male means this is a female and this is a male part. You have anther and filament. So this anther is to be removed. Can you see here? We are removing anther by the process, right? By the process, this process of removal of anther, okay, where the anthers are removed, this process is known as emasculation. What it is known as? It is known as emasculation. In your textbooks they have not explained not in much detail, but I am explaining you because you should know the meaning. After the process of emasculation, what will happen? The third process, like the third step is of, you have to bag it. Why we are doing bagging? Bagging means what? See, I will show you. See here, the flower. See, this is a flower and the flower is bagged. Okay, if you can see that there is a bag. Okay, which bag they have used? In any polythene bag they have used to cover. Now why we are doing this process of bagging? To avoid any contamination to the flower. अगर फ्लावर में कुछ भी अनवांटेड सामान जाएगा तो मतलब नहीं है, बिकॉज ये आर्टिफिशियल हाइब्रिडाइजेशन है, ये रैंडम फिनोमेना जैसे होता है नेचर में वैसा नहीं करवाना है हमें. सो because of this reason, like we want some desired product, we want desired pollen to fall on the stigma. Due to this reason we are doing bagging. Okay, so this is the process of bagging. After bagging, the fourth step is hand pollination. Hand pollination. In this, what you have to do, you have to dust the pollen, means you have to open the bag, you have to remove the bag and you have to dust the pollen, and again what you will do, rebagging. Again you have to put the bag, rebagging. Okay, so this is the step, and these are the two images which I had shown you. Okay, after this, last step, sixth step is tagging. Now what is tagging means which pollen you had added, what is the name of the plant, when you added, okay, means when you dusted the pollen, date, day, location, who dusted the pollen. These all information is there in a tag. If you can see in this image, there is some tag here. Okay, so you have to write all the information which I told. Okay, like name and all, name of the plant and everything. Okay, this is a tagging. You can see these steps also, the same thing written in different language, but it is same. This is the thing which I have written, more simplified version I have given you. You can see this step also and you can understand, it's not that difficult. So this was about artificial hybridization. Now we will come towards our next topic of today's class, that is double fertilization. What are we going to study? Double fertilization. In the last class we have studied that okay, I will draw the diagram, that there was a stigma, there was style, right, there was ovary. What was happening here? There was ovule and the pollen had germinated and it was traveling towards stigma to style, and it came to the nearby the ovary, and this way the pollen pistil interaction, and that too chemical interaction was done. Now the entry of pollen tube, if you can see here, the pollen tube have entered, but now from where it will enter in the ovule. Okay, so there are three positions from where it can enter. Okay, so entry of pollen tube. So pollen tube can enter from three places. How many places? Three places. First place from where the pollen tube can enter is known as porogamy. Poro means from a pore. Now which is this pore? The pore is known as micropyle. Okay, it is known as micropyle. So in ovule there is a presence of micropyle, pilar end. Okay, so from micropyle it can enter. Okay, so pollen tube will go towards the micropilar end, or I will write simply micropyle. Okay, this is the first entry. Now the second place from where it can enter, the second place is chalaza. Okay, so the process of entry from chalaza means the process means this process, like from, if the pollen tube enters from the chalaza, so we say it as chalazogamy. Chalazogamy. Okay, and if suppose it enters from integuments, we say it as mesogamy. Okay, integuments. Mesogamy is for integuments. Okay, and chalazogamy is for chalaza. Okay, I will not write from gray, it's not visible properly. Okay, so these are the three types, like from where it will enter. First is porogamy. I will show the image also. It is, and in the porogamy it is entering from the micropylar end. In the chalazogamy it is entering from chalaza. In the mesogamy it is entering from integument. Okay, so this was the entry of pollen tube. Now I will show the diagram. If you can see here, in the micropylar end, I will draw this pollen tube, pink color. Can you see this pink color? It is entering from micropyle. So we are saying it as porogamy. Okay. Again I will draw it. See here. Can you see that it is entering from chalaza? Okay. Here it is entering from chalaza. So we say it as what we say. See, it is coming from here. See, it is coming from here. It's not coming from outside. ऐसे बाहर से नहीं आ रहा है. अगर बाहर से आता तो नीचे से होता तो we would have told it as porogamy. But see, it is coming from here. Okay. So this is chalazogamy. I will draw it again to show you properly, because all the black and white image you will not understand. See this pink color line which I have drawn. This is chalazogamy. If it is coming from the integuments, the pollen tube is entering from the integuments, then we say it as mesogamy. Okay, so we have total 3 types. You should know these types, right? Okay, so this is about the types. Now we will come towards the double fertilization. See, we remember the structure of, the structure of ovule, embryo sac. Okay, so first of all, double fertilization is a complex event. Double fertilization is a complex event. First time double fertilization was given by a scientist known as, the name is Navas. Okay, you should know this. He first time observed double fertilization, which is a complex process in angiosperm. Complex process where it observed in angiosperms, that is your flowering plants. Who observed this? It was observed by, this phenomenon was given by Navas. The entire name is S. Navas. Okay. So this person gave, and in which plant, which family. So he gave this in liliaceous plants. Okay, liliaceous plants. And in liliaceous plants, he actually studied lilium and fritillaria. Lilium and fritillaria in these 2 plants. Okay, so I wrote liliaceous plants. In this group of plants he found it out. Next, now if we come towards the further part, see, we know that, I will come in the next page. See, we know that pollen grain is there. Pollen grain came from where? It came from the anther, and it came by the process of pollination. It came to It landed on the stigma, and from stigma the pollen pistil interaction begin. And in this pollen pistil interaction, what happened? The pollen grain germinated. Pollen grain germinated. ठीक है, it germinated. Then what happened? In this germination, the two important things occurred. First important thing, that pollen tube was formed, and second important thing, two male gametes were transferred. Okay, so these were the two important things which occurred. Now pollen tube is actually taking two male gametes. I had shown you the image. Let's see one image. See here. Can you see that pollen tube is taking sperm nuclei, means what? Male gametes. Okay, this is nothing but male gametes. So pollen tube is taking, it's on the stigma, it's taking, you can see it will cross style, it will go towards ovary. It can enter in either from integument or either from micropyle or chalazo end, and it will go towards egg cell, means it will go to the egg cell. Okay, so we have two sperm cell. Now let's come to the part. So I explained this picture, like this concept. Let's move forward. So now what will happen? I told that there will be some chemical reactions. Very important point. What will be there? There will be chemical interactions. Okay. Now pollen tube came inside. It is near ovary. Can you see here? It is near ovary. Now what happens near ovary? Pollen tube. Now this pollen tube actually goes towards synergids. Okay, and synergids, from synergids it absorbs. What it does? It absorbs water. Whatever fluid things it will get, chemicals and all, it will absorb. Okay, in short, it will absorb water. Okay, after the absorption of water, what actually occurs is this, like, see, if pollen tube is going towards the synergid and it's carrying two male gametes, how many? Two male gametes. Okay, and here what will happen? Here it will, here if you will see the diagram, I will show the diagram also. Okay, see, here it's going here, and here you have two synergids, and in middle you have egg cell. So, and even you have fimbriae here. So fimbriae is here to show the direction to the pollen tube. Okay, it will show direction to the pollen tube, and it's present near the synergid. So fimbriae is present. We know this already. We have studied this in when we were studying the female gametophyte. Okay, so entry will occur, suppose its entry is occurring from micropylar end. So we spoke about the three types. Micropyle, if it is entering from micropyle, so we will say it as porogamy, chalazogamy and mesogamy. Okay, if it is entering from integument, so we will say mesogamy. Okay, these are two synergids, and here you have egg cell. So this is M1, means gamete number 1. Okay, this is gamete number 1, and you have gamete number 2. Now what will happen? The gamete number 1 fuses with the egg. Okay, before fusing, okay, before the fusion, the pollen tube must burst, right? Before the fusion, pollen tube must burst. Means what? The male gametes should come out of the pollen tube. So here what is happening? Pollen tube, like, it absorbed It absorbs, absorbs water, H2O, from synergids. Whatever fluid it got, it absorbed. Okay, from synergids. And then what will happen? It absorbed so much that it became swollen, and finally it started bursted. Like it actually bursted. The moment it bursted, it released two male gametes. And now the story about the two male gametes, like what these two male gametes actually did. These two male gametes, obviously now these two are non-motile male gametes. Okay, so it got bursted. The moment the pollen tube got bursted, very important here, the pollen tube is getting bursted because it actually took some water. So because of the absorption of the water, the pollen tube bursted, or you can say ruptured. Okay, you can say that pollen tube got ruptured and these two male gametes got released. The moment these two male gametes got released, and it will get released, okay, these are two male gametes which are non-motile, hence why what was the purpose of this pollen tube? I hope you understood, because two male gametes were non-motile. So it will go inside here, and we have here secondary nucleus also. If you remember the diagram, so we have polar nuclei, which is also known as secondary nuclei. So now the two male gametes have entered here. Suppose these are two male gametes, M1 and M2, male gametes, so I have referred it. So this M1, okay, M1 went here and it fused with egg. So this is M1, means male gamete, first male gamete, okay, which was haploid. It went and fused with the female gamete. Okay, this is a female gamete. They both fused. This fusion is nothing but it is known as fertilization. What it is known as? It is known as fertilization. Here fertilization occurred, and N plus N is nothing but 2N. Okay, and this is where the formation of zygote occurred. This is the first thing. Again I am repeating, the first male gamete, okay, first male gamete fused with the egg. Egg is also haploid, male gamete is also haploid, and we got diploid zygote. Okay, we got diploid zygote. So here the ploidy is restored. We need a plant is a diploid, like, so the ploidy got restored. This process, the process where the one female gamete fuses with the one male gamete, this is known as fertilization. Okay, and another term for this is known as syngamy. What it is known as? Syngamy. Syngamy means where male and female fuse to form diploid zygote. Okay, how is the zygote? Zygote is diploid. It is diploid, and the male and the female gamete are haploids, N. This is a process. This is the information about syngamy. Further, I told that there are two male gametes. Now what will happen of M2? Second male gamete fused with the polar nuclei. You can say polar nuclei, or you can also say it as secondary nucleus. What you can say secondary nucleus. And this is a haploid, this is a diploid structure. You should know, and male gamete is a haploid structure. If you will see here, the polar nuclei, actually it is not fused. It is having one cell wall, and inside you have two nucleus. So this nucleus is haploid and this nucleus is haploid, so we are saying it as diploid. So polar cell of polar nuclei is a diploid structure. So here is there is a fusion of haploid and diploid, which you will get, like, which will give you a triploid structure. What you will get? Triploid. 1 plus 2 is 3. So here you will get a PEN. What is PEN? PEN means primary endosperm nucleus. Okay, अभी जाइगोट तो बन गया, ठीक है, पर जाइगोट को खाना भी तो चाहिए. ये जाइगोट तो बन गया, ठीक है, आई एग्री कि जाइगोट बना है, दिस इज द जाइगोट, but zygote needs food. What it needs? It needs food. खाना चाहिए उससे, right? Developing zygote needs food. So this food is provided by PEN, that is primary endosperm nucleus. Who provides primary endosperm nucleus? This is actually kitchen of zygote. What it is? It is a kitchen of zygote. This process of formation of primary endosperm nucleus, this process is known as triple fusion. What it is known as? It is known as triple fusion. Now why it is known as triple fusion? It is known as triple fusion because your haploid male gamete fuses with a diploid polar nuclei to give a triploid structure. So we say it as triple fusion. What we say? We say it as triple fusion. So this is, there are two fusion occurring. First, male gamete is fusing with the egg. So this was first fertilization, you can say, first fertilization, and in second time, like the second male gamete, if you can see here, the second male gamete, it is fusing with the secondary nucleus, or you can say polar nucleus. So this is second time fertilization. What we say? We can say this is second time fertilization. What it is? It is a second time fertilization. So one and second fertilization. So there are total 2 time fertilization. So we say it as double fertilization. Why double fertilization? Because male gamete first was fusing with egg, okay, egg, and male gamete second was fusing with polar nucleus. So here you got diploid structure zygote, Z means zygote. Here you got triploid structure, that is PEN, pen means primary endospermic nucleus. Now I will tell you one important thing which is very, very, very important. If you can see here, male gamete and female gamete are fusing to give you what? To give you zygote. Fine. So this process, this first fertilization is also known as generative fertilization, because here you are getting new generation. So this is generative fertilization. Whereas when second male gamete what was fusing with the secondary, or you can say polar nuclei, here you are not getting zygote, here you are getting kitchen. This kitchen is primary endospermic nucleus. This is actually nutrition for zygote. What is this? This is a nutrition for zygote. Okay, this is a nutrition for zygote. So this is not a new zygote, or this will not form any embryo or something. So we can't say that it will give any generation. Here first the first fertilization was giving new generation. The first fertilization will give new generation. But the second fertilization won't give any generation. So the second fertilization is known as vegetative fertilization. What it is known as? Vegetative fertilization. These are the very important terms you should know. What is for generative fertilization and what is vegetative fertilization? Okay, so since there are two times fertilization are occurring because both the male gametes are participating. Why double fertilization? Because both male gametes are participating in fusion. Both male gametes are participating. So because of this reason we say it as double fertilization, because both male gametes are participating. Okay, so here we have the topic of double fertilization. Whatever were the important points I told you, okay, I will just go through the thing. I will just show you. See if you can see the release of sperm cells. Here they are showing you the release of sperm cells, and you can see the process of double fertilization. Here you are getting zygote. Here you are getting endosperm. Endosperm is nothing but, you can say, kitchen of zygote. Okay, that is nothing but, you can say, nutrition for zygote. Nutrition for zygote. Okay, next, if you will see, here also I have shown the same diagram. You can see this was a one ovule was like, they are zooming one ovule from here, and you can see the diagram. And another diagram is again showing the same thing, that formation of zygote and all. Now I will just show you this. So this, in the part 8 we first studied about the artificial hybridization. Okay, and I told you the entire process, the steps also I taught you. Then we came towards the pictures of the steps. Then we studied about double fertilization event, which is very, very important in terms of your exam point of view. So this is a very important topic. You should literally write the notes properly for this very important topic. And then I taught about the 3 types of pollen tube entry. Then we spoke about double fertilization in detail. I taught you one small flowchart. Then this process I taught, how the pollen tube, like from pollen tube, how male gametes are coming out. So they are coming out because they have, the pollen tube had like it is absorbing the water from the synergids. And I told the role of filiform apparatus here. Okay, what is the role? It is showing the direction to the pollen tube. So this is the role of filiform apparatus. I will write it here, filiform apparatus. Okay, then next you can see that the fertilization I have taught. Why double fertilization, what is generative, what is vegetative fertilization. And so we can end for today's lecture. In the next lecture we will see the significance of double fertilization, and we will start with the development of endosperm. So thank you for watching this lecture. Do comment below if you have any doubts. Thank you for watching.